You're listening to LeadAI.dev, your daily updates on AI development. Today's topic is MolMem. Memory-augmented AI cuts drug discovery costs by 90%. Here's a quick overview. Breakthrough MolMem framework dramatically reduces molecular optimization costs through memory-augmented reinforcement learning, making drug discovery accessible to smaller research teams. Now, let's ask our analyst about these developments. What's new? MolMem framework transforms molecular optimization with memory-augmented AI.
Researchers have unveiled MolMem, a groundbreaking memory-augmented agentic reinforcement learning framework that addresses the critical sample efficiency challenge in molecular optimization. Published in ArcSiv, the system reduces oracle evaluation requirements by up to 90% compared to traditional trial and error approaches, potentially transforming drug discovery economics. The framework combines episodic memory mechanisms with reinforcement learning agents to iteratively refine lead compounds while maintaining structural similarity to original molecules. Unlike existing methods that require hundreds of expensive oracle calls, MolMem learns from previous molecular transformations stored in memory, enabling more efficient exploration of chemical space. The technical architecture centers on an agentic system that maintains detailed episodic memories of successful molecular modifications. Each memory entry contains molecular fingerprints, transformation actions, property improvements, and structural constraints. The reinforcement learning agent queries this memory during optimization to identify promising modification patterns without requiring new oracle evaluations. The system employs attention mechanisms to retrieve relevant historical examples based on molecular similarity and property targets. This approach bridges the gap between computationally expensive ab initio methods and less accurate template-based approaches. Previous molecular optimization methods faced a fundamental trade-off between sample efficiency and exploration capability. Template-based approaches reused familiar patterns but struggled with novel chemical spaces, while reinforcement learning methods required extensive oracle budgets for adequate performance. MolMem eliminates this trade-off by creating a dynamic knowledge base that grows with each optimization episode. The memory system enables transfer learning across different molecular targets, allowing insights from one optimization campaign to accelerate subsequent projects. This represents a paradigm shift from isolated optimization runs to cumulative learning across molecular discovery programs. Reduces oracle evaluation requirements by 90% compared to baseline reinforcement learning methods, implements episodic memory system storing molecular fingerprints, actions, and outcomes, employs attention-based retrieval for identifying relevant historical transformations, enables transfer learning across different molecular optimization targets, maintains structural similarity constraints while optimizing multiple molecular properties.
Thank you for that analysis. Let's move on. Who benefits from memory-augmented molecular optimization?
Pharmaceutical companies with limited computational budgets represent the primary beneficiaries of MolMem's efficiency gains. Mid-sized biotech firms spending $50,000,200,000 annually on molecular modeling can now achieve results previously requiring million-dollar budgets. Research teams working on rare disease therapeutics, where small patient populations limit commercial viability, gain access to sophisticated optimization tools without prohibitive costs. Academic drug discovery programs operating under grant constraints can now pursue more ambitious molecular targets. The framework particularly benefits organizations optimizing multiple properties simultaneously, such as potency, selectivity, and AD-MET characteristics, where traditional methods require exponentially more evaluations. Contract research organizations, CROs, and computational chemistry consultants can leverage MolMem to deliver faster turnaround times while reducing client costs. The memory system enables these organizations to build proprietary knowledge bases across client projects, creating competitive advantages through accumulated molecular insights. Artificial intelligence teams developing drug discovery platforms can integrate MolMem as a core optimization engine, reducing infrastructure requirements while improving client outcomes. Research institutions collaborating across multiple projects benefit from shared memory systems that accelerate discovery across different therapeutic areas. Organizations should delay adoption if they lack sufficient historical molecular data to populate initial memory systems effectively, as the framework requires baseline optimization episodes to demonstrate full benefits. Teams working exclusively on well-established molecular scaffolds with abundant literature precedents may find traditional template-based approaches more immediately practical. Companies requiring real-time optimization for high-throughput screening applications should wait for optimized inference implementations designed for production environments. Mid-sized biotech firms with $50K200K annual computational chemistry budgets, academic research groups pursuing multi-objective molecular optimization CROs, seeking competitive advantages through accumulated molecular knowledge, AI platform developers building drug discovery solutions.
Great insight, really helpful context. How to get started? Implementing MolMem for molecular optimization?
Implementation begins with establishing the computational environment and molecular representation systems. Install required dependencies, including RDKit for molecular manipulation, PyTorch for neural networks, and specialized cheminformatics libraries. Configure molecular fingerprinting methods such as ECFP or Morgan fingerprints to ensure consistent molecular representations across memory entries. Establish Oracle evaluation interfaces for your target molecular properties, whether through quantum chemistry calculations, machine learning models, or experimental assays. Create data pipelines for pre-processing molecular structures and property measurements into standardized formats compatible with the memory system. Initialize the episodic memory database with existing molecular optimization data if available, or begin with empty memory for new projects. Configure memory retrieval parameters, including similarity thresholds, attention weights, and maximum memory size based on computational resources. Set up the reinforcement learning environment with appropriate action spaces for molecular modifications, such as atom substitutions, bond formations, or functional group additions. Define reward functions that balance property improvements against structural similarity constraints. Implement safety checks to prevent generation of chemically invalid structures during optimization episodes. Launch initial optimization runs with conservative exploration parameters to populate the memory system with high-quality examples. Monitor memory utilization and retrieval patterns to optimize similarity thresholds and attention mechanisms. Gradually increase exploration as memory coverage improves, allowing the agent to attempt more ambitious molecular transformations. Implement checkpointing systems to preserve memory states across optimization campaigns, enabling transfer learning for future projects. Validate optimization results through independent property calculations or experimental verification when possible. Install RDKit, PyTorch, and cheminformatics dependencies for molecular processing, configure ECFP or Morgan fingerprints for consistent molecular representations, initialize episodic memory with existing data or empty database for new projects, set similarity thresholds and attention weights based on computational resources, implement safety checks, preventing chemically invalid structure generation, enable checkpointing for memory preservation across optimization campaigns.
That's a very interesting point. Tell us about competitive context. MolMem versus traditional molecular optimization methods.
MolMem significantly outperforms traditional reinforcement learning approaches like reinvent and ChemTS in sample efficiency metrics. While reinvent typically requires 1-0-0-0-0-5-0-0-0-0 Oracle evaluations for convergence, MolMem achieves comparable results with 1-0-0-0-5-0-0-0 evaluations through memory-guided exploration. Compared to genetic algorithms like NSGA, MolMem demonstrates superior handling of multi-objective optimization scenarios by learning from successful trade-off solutions stored in memory. Template-based methods like RecApp and Bricks offer faster initial results, but plateau quickly due to limited exploration capabilities, whereas MolMem continues improving through accumulated experience. The framework also surpasses graph-based approaches like GraphQA in maintaining structural coherence while exploring novel chemical modifications. Key advantages include cumulative learning across optimization episodes, enabling transfer of molecular insights between projects targeting different properties or scaffolds. The attention-based memory retrieval system provides interpretable rationale for molecular modifications, unlike black box neural approaches. Memory-augmented exploration reduces the likelihood of getting trapped in local optima that plague traditional reinforcement learning methods. The framework scales efficiently with available computational resources, automatically adjusting memory size and retrieval complexity based on hardware constraints. Integration capabilities exceed competing methods through modular architecture supporting various Oracle types and molecular representations. Limitations include dependency on sufficient historical data for optimal performance, making cold start scenarios less effective than methods designed for limited data regimes. Memory storage requirements grow linearly with optimization episodes, potentially creating scalability challenges for very large molecular libraries. The framework may overfit to historical patterns when memory contains biased or low-quality examples, requiring careful curation of stored experiences. Real-time inference speeds currently lag specialized template-based methods optimized for high-throughput applications. Achieves 8.0.
90% reduction in Oracle evaluations compared to reInvent, and ChemTS outperforms genetic algorithms in multi-objective optimization scenarios, provides interpretable molecular modification rationale through attention mechanism scales efficiently across different computational resource constraints.
Good analysis. Let's continue. What's next? Future developments in memory-augmented molecular discovery?
Development roadmaps indicate expansion toward multimodal memory systems incorporating experimental data, literature knowledge, and synthetic accessibility information alongside molecular structures. Researchers are exploring federated learning approaches enabling secure memory sharing across pharmaceutical organizations while preserving proprietary information. Advanced memory architectures under development include hierarchical storage systems that maintain both episode-specific and general molecular transformation patterns. Integration with large language models trained on chemical literature promises to enhance memory retrieval through natural language queries about molecular properties and transformations. Automated memory curation systems will identify and remove low quality or outdated examples to maintain memory effectiveness over time. The broader molecular AI ecosystem shows increasing adoption of memory augmented approaches across related applications, including retrosynthetic planning, reaction prediction, and materials discovery. Major pharmaceutical companies are investing in memory-based optimization platforms as core infrastructure for drug discovery pipelines. Cloud-based implementations will democratize access to sophisticated molecular optimization capabilities for smaller research organizations. Integration with robotic synthesis platforms enables closed-loop optimization, where memory systems guide both computational design and experimental validation cycles. Long-term implications suggest fundamental shifts in drug discovery economics as memory-augmented systems reduce barriers to molecular optimization. The technology may accelerate development of personalized therapeutics by enabling rapid optimization for specific patient populations or rare genetic variants. Academic industry collaborations will likely emerge around shared memory systems containing anonymized molecular optimization experiences. Regulatory frameworks may evolve to accommodate AI-designed molecules with transparent optimization histories stored in memory systems. Multimodal memory systems incorporating experimental data and literature knowledge, federated learning approaches for secure memory sharing across organizations, integration with large language models for natural language molecular queries, cloud-based platforms, democratizing access to sophisticated optimization tools.
Great analysis. So, what are the key takeaways from all of this?
MolMem reduces molecular optimization costs by 90% through memory-augmented reinforcement learning, making advanced drug discovery accessible to smaller research teams. Episodic memory systems enable transfer learning across molecular targets, allowing insights from one optimization to accelerate subsequent projects. Implementation requires RDKit, PyTorch, and proper molecular fingerprinting setup, with gradual exploration increases as memory coverage improves. The framework outperforms traditional methods in sample efficiency while providing interpretable molecular modification rationale through attention mechanisms.
And what market signals should our listeners watch for?
Democratization of drug discovery. MolMem's 90% reduction in computational requirements fundamentally alters drug discovery economics, enabling mid-sized biotech firms and academic institutions to pursue sophisticated molecular optimization previously reserved for pharmaceutical giants. This democratization accelerates therapeutic development for rare diseases and personalized medicine applications where traditional approaches prove economically unfeasible. The technology shifts competitive advantages from computational resources to data quality and memory curation strategies, potentially disrupting established pharmaceutical research hierarchies. Memory-first AI architecture adoption. The success of memory-augmented molecular optimization signals broader industry movement toward memory-first AI architectures across scientific applications. Organizations are recognizing that accumulated experiential knowledge often outweighs raw computational power for complex optimization problems. This trend extends beyond drug discovery into materials science, catalyst design, and chemical synthesis planning. Companies investing in memory infrastructure and curation capabilities gain sustainable competitive advantages through accumulated domain expertise encoded in retrievable formats. Collaborative knowledge ecosystem emergence. MolMem's transfer learning capabilities drive development of collaborative molecular ecosystems where organizations share anonymized optimization experiences. This creates network effects where participation value increases with ecosystem size, potentially leading to industry-wide consortiums for molecular discovery. Early adopters establishing high-quality memory systems become attractive collaboration partners, while organizations remaining isolated face increasing disadvantages. The shift towards shared molecular intelligence may require new intellectual property frameworks and data governance models.
What should developers and operators do with this information?
Implement MolMem pilot projects within 3 months for teams with existing molecular optimization budgets exceeding $50,000 annually. Establish memory curation protocols immediately to ensure high-quality episodic storage from initial optimization campaigns. Begin federated learning partnerships with complementary research organizations within 6 months to accelerate memory system population. Integrate MolMem with existing computational chemistry workflows before Q3 2024 to capture optimization history for memory initialization.
Thank you for listening to LeadAI.dev. Stay tuned for our next daily updates in this rapidly emerging market.